All right, I've made a second part to this urinary system PowerPoint because my first video got cut off somewhere around where we were introducing the parts of the nephron. So I've sketched the nephron back on the board, and we've got some blood vessels that surround the nephron. So maybe we can start by labeling that as a review, and then we'll jump back into the parts of the nephron. We know that this blood vessel that is feeding blood to the particular capillary bed that the nephron is biting onto, this blood vessel we called the afferent arteriole. It was delivering blood to that tangled bed of capillaries that we called the glomerulus. And that glomerulus, if we were a red blood cell moving through the glomerulus, we would end up in the efferent arteriole. Maybe for reference, I can put these little arrows that show afferent arteriole is delivering blood to the glomerulus, and efferent arteriole is taking blood away from the glomerulus. This efferent arteriole, if we keep following it out, this blood vessel is going to continue and it's going to get called a different name. When it's around these two portions of the nephron that we're going to call the proximal convoluted tubule and distal convoluted tubule, then that blood vessel is going to get called the paratubular capillary. So let's label that real quick. So paratubular capillaries would be here and here. We're referring to the blood vessels that are around the proximal and distal convoluted tubules of the nephron. And then there's the other blood vessel that we can see that drops down around this loop of Henle that we are about to label. This blood vessel that's kind of dropping around the loop of Henle we call the vasa recta. There it is. And if we were to see where the rest of this vasa recta leads to, you can see that we've kind of gone from an artery through this capillary and now to a vein you will end. This vasa recta is going to continue and make its way over here and merge with these vertical blood vessels. Perhaps we could label those. I've got an artery in red and a vein in blue. We're looking at the systemic circuit. But if we just look at the arteries real quick, we discussed this afferent arteriole taking blood to the glomerulus. This afferent arteriole is receiving blood from this cortical radiate artery. Let's label this red structure here our cortical radiate artery. And you can see right next to it in blue there is a cortical radiate vein. The cortical radiate artery and cortical radiate vein also go by another name called the interlobular artery or interlobular vein. I like to use cortical radiate because it describes that these are the blood vessels that radiate out into the cortex. These cortical radiate arteries are going to receive blood from these blood vessels that are going to be running right along the base of one of the renal pyramids that would be in this triangular shaped area. We called those blood vessels the arcuate arteries and arcuate veins. In fact, let me just relabel this real quick to give myself a little more space. These blood vessels that we can see running along the base of the pyramids, we call those arcuate arteries and veins. And we see the blood vessels that are running along the sides of the pyramids. We call those interlobar arteries and veins. The term interlobar artery and vein sounds an awful lot like interlobular. So instead of using the term interlobular to describe cortical radiate arteries and veins, I use the cortical radiate term because it's a lot different sounding than interlobar artery and vein. So again, interlobar between the pyramids, arcuate artery along the base of the pyramids, cortical radiate arteries radiate out into the cortex. Branching off of that cortical radiate artery, we have our afferent arteriole that leads into the glomerulus. As we move away from the glomerulus by way of this efferent arteriole, we end up either in the peritubular capillaries or the vasa recta, depending on where we're at. And this vasa recta, these veins are going to end up making their way right back into the cortical radiate vein. And then the veins are going to make their way right back out of the kidney. In the first part of this PowerPoint, we traced the blood flow through the kidney. And in the second part, we really talk about the parts of the nephron. So now that we've reviewed our blood flow through the kidney, let's label the parts of the nephron so that we can talk about what each segment does. The first part that we can label is this Pac-Man looking structure. We're going to call it Bowman's capsule. Bowman's capsule is also called the glomerular capsule. It surrounds this glomerulus. And if we move away from Bowman's capsule, the first part of this squiggly lines here, this first series of squiggly lines is going to represent our proximal convoluted tubule. We'll see as we continue to move through the nephron, it starts with the Bowman's capsule, then we move through the proximal convoluted tubule and down into this long loop. This is called the loop of Henle. And it has a descending limb. That's the first part. We're going to see filtrate travel down this descending limb. It does a little sharp turn at the base of this loop, and then the filtrate is going to make its way up. Notice how there is a thin segment and a thick segment of this ascending limb. We'll talk about a distinction between the two different parts of the ascending limb. But this ascending limb of the loop of Henle is going to run into our last series of squiggly lines. In this case, they represent the distal convoluted tubule. And the distal convoluted tubule, we can see running into this vertical tube that I've drawn. I'll just label this last vertical tube as the collecting duct. In this collecting duct, what we can see is that it is shared by multiple nephrons. So I've drawn a distal convoluted tubule that is leading into the collecting duct, but the other little thing that I've drawn on here, this is also a distal convoluted tubule of a completely different nephron that's dropping its filtrate into this collecting duct. So the collecting ducts are hooked up to a bunch of different nephrons, not just the one that we've drawn, and they're all dropping their filtrate into this collecting duct. It's going to end up dropping down into what we looked at on the lab model as a renal papillae. The tip of one of these pyramids is what secretes filtrate down into a minor calyx. So anything that makes its way out of this collecting duct is going to end up in the urine. And urine is going to be mostly things like sodium ions and water. But depending on things like if your blood is acidic, we're going to see that the urine is also going to be acidic. That's one of the ways that the kidneys try and compensate for whatever blood issue you might be facing. If your blood is too acidic, we'll see that the kidneys are going to remove those hydrogen ions and put them in the urine. So we'll see the urine is acidic. The opposite is true if your blood is too alkaline, if it has too many of these bicarbonate ions in it, then we're going to see that the urine is also going to be too alkaline. Quick side note, if the kidneys are regulating hydrogen ion concentrations or this bicarbonate ion concentration, one of the side effects that it is also going to be holding on to or getting rid of is potassium ions. So depending on what's going on, there may be varying levels of potassium in the urine. In fact, we'll need to look back at the beginning of this nephron to see what might end up in the urine. Basically, sodium, water, and then some of the other ions are going to vary depending on what cells are active along the nephron. And we might as well throw those cells in there real quick. It's 3 different types of cells we need to know. I'll try and squeeze them in here in an orange color. And these three cells that we need to know are the cells that are influenced by hormones. They're found along the distal convoluted tubule and this collecting duct. Just to save space, I'll draw them along the collecting duct. And I have just enough room to give them a name. We are likely to erase this diagram in just a second, and we'll draw a more zoomed in picture of what's going on right here between the between the collecting duct, these cells that we're about to draw, and vasa recta or paratubular capillaries. I'll put the first cell in here, and let's call this one a principal cell. That principal cell is going to be one of these cells that is going to be regulating things like water and sodium. So I'll put an ion in there of sodium and a water molecule, just indicating that this cell is going to help reclaim things like water or sodium. We'll talk about the hormones that trigger it to do that. But let's introduce the other two cells that play a role. There is, just draw them both on here. The first one is going to be what we call a type A intercalated cell. Type A intercalated cell, and the second one is a type B intercalated cell. These principal cells, type A and type B intercalated cells, we can see line the distal convoluted tubule and collecting duct. The latter two there, the intercalated cell, type A and type B, are going to play a role in helping the blood regulate whether the blood is too acidic or too basic. Maybe that's one way that you can easily remember these. Type A intercalated cell is going to kick into gear if the blood is too acidic. Maybe combine the type A with blood being too acidic. We're going to see that what this type A cell is going to do is it's going to pull hydrogen ions out of the bloodstream that's too acidic, and it's going to get rid of those hydrogen ions into the urine. That's one of the ways that we could end up with extra hydrogen ions in the urine. Like I just said, if the blood is too acidic, the urine is also going to be acidic because these intercalated cells are getting rid of hydrogen ions. We'll talk about more of exactly how it does that in just a second. There's more to the story. But introducing these type B intercalated cells, think that these kick into gear if the blood is too basic or too alkaline. In that case, there's not enough of those hydrogen ions in the bloodstream. The trick that these type B intercalated cells can do is that they can take hydrogen ions and put them directly into the bloodstream. And that's going to help compensate for the blood not having enough of those hydrogen ions. We're going to see there's also more to the story than that. In order to get rid of one of these hydrogen ions, we're going to see the cell, maybe the other side to that, just to throw in that real quick. The cell is going to have to pick up a potassium ion. And this potassium ion is going to get lost in the urine. Like we said, sometimes there are potassium ions in here. So depending on what cells are active, type A intercalated cells, type B intercalated cells, or even these principal cells, it's going to affect what ends up down here in the urine. So we'll jump to this in just a second, but before I erase this diagram, let's take a quick journey back to the start of this nephron just to remind us of what's going on. And I know this is all up here on the PowerPoint in front of me. The first thing that we can see occurring right there in Bowman's capsule is filtration. We described this last time right before the video cut out. We said that there are these tiny little pores in the capillaries found in the glomerulus. And if molecules are small enough to fit through those pores, they're just going to squeeze out and find themselves in Bowman's capsule. This is what we refer to as filtration. Kind of things making their way out of the capillaries and into Bowman's capsule. We said that if you're small enough to fit through those pores, you're going to end up down here in Bowman's capsule. That's going to go for some of the things that we want to get rid of, as well as some of the things that we want to keep. So water molecules are going to be small enough to fit through. We're going to see ions are small enough to fit through. So ions like sodium ions or potassium ions, hydrogen ions. Even things like glucose molecules are small enough to squeeze through. I'll draw those as a little hexagon shape. And then we could also include waste like urea is also going to make its way out of those little filtration slits and into Bowman's capsule. So it's just selecting based on size. If you're small enough, then you're going to make your way out of the glomerulus and into Bowman's capsule. But some of that stuff we don't actually want to end up down here in the urine. So there are parts of the nephron whose job it is to reclaim some of the things that we would like to reuse. For example, as this filtrate is passing through the proximal convoluted tubule, maybe the key thing that we can think about for the proximal convoluted tubule is it's the only part of the nephron that has a chance to reclaim organic compounds, any glucose, for example. The only place that this is going to get pulled back into the bloodstream is right here in the proximal convoluted tubule. There are certain glucose transporters that are found only on the proximal convoluted tubule. They're going to let these glucose molecules cross. The glucose molecule ends up kind of temporarily in this extracellular fluid, but then it's picked up by the peritubular capillaries. This is what we consider reabsorption. If we go back, remember this glucose was already in the bloodstream. It just kind of popped out temporarily, and then it's pulled right back into the bloodstream. So filtration is when it leaves the bloodstream, and then when it's pulled back into the bloodstream, we might as well write that down as reabsorption. As opposed to just absorption of nutrients that happened in the small intestines. We talked about that last time in the digestive system. This is reabsorption, again, because it was already in the bloodstream to begin with. It just popped out temporarily. So the notable thing about the proximal convoluted tubule is that by the time the filtrate makes its way through this proximal convoluted tubule, any glucose molecules that had been filtered out are all going to be picked back up by those peritubular capillaries. 100% of glucose is going to get reclaimed in nephrons that are working properly. It should be noted that people that are diabetic have so much glucose in their bloodstream, it's at such high loads that it overwhelms these glucose transporters and it's not able to pick up all of the glucose. So inevitably, if some of the glucose ends up past the proximal convoluted tubule, there's nothing left in the nephron that's going to reclaim it. It's going to end up down here in the urine, which is why they were able to detect the first diabetics, because bees were attracted to their urine. There was sugar in their urine, and somebody actually tasted the urine to find out that it was sweet tasting. So then there was a race to figure out how did that glucose get into the urine, and it led to discovery of the nephron and how parts of it work. So all of the glucose gets reclaimed in the first part of this proximal convoluted tubule. Most of the water, so we're going to see there are channels for water molecules to be pulled out of this proximal convoluted tubule. That water ends up in that extracellular fluid temporarily and then is pulled right back into that peritubular capillary. About 99% of the water that is filtered out in Bowman's capsule is going to get picked back up by these peritubular capillary beds as it is reabsorbed through this proximal convoluted tubule. Sodium, where's my sodium color? We're going to see sodium is mostly going to be reabsorbed here by the proximal convoluted tubule. It's going to get picked right back up by those peritubular capillaries. The things that don't get picked up are the waste like urea. And it turns out some of the sodium ions and some of the water molecules are going to still make their way down this loop of Henle. So since I'm a little bit crowded for space, let me try this. I'll use my blue dots to represent water molecules that did not get picked up by that proximal convoluted tubule. We know some water molecules are going to make it out in the urine. I'll use this green dot to represent my sodium ions. We know not all the sodium got picked up in the proximal convoluted tubule. Some of it's going to end up down there in the urine, so we have that moving down the tube. And then I'll use an orange color to represent the waste, the urea, that's also not reabsorbed by the proximal convoluted tubule. So we've got all this filtrate, some water, some ions, some waste that didn't get reclaimed in the proximal convoluted tubule. It's going to make its way down this loop of Henle. So here's where we need to talk about this descending limb, the first part of this loop of Henle. I won't be able to draw it on here, but I need to mention that there are special cells that line this descending limb of the loop of Henle that are permeable to water molecules and nothing else. So water molecules, we're going to see, are going to end up going out of this descending limb. What happens is that as this filtrate is pulled further and further down this loop of Henle, water molecules are extracted. They are pulled out of this descending loop of Henle, and they're going to get picked right back up by this blood vessel, this vasa recta. We said anything that comes out of the proximal convoluted tubule or distal convoluted tubule are going to get reclaimed by these paratubular capillaries, but we'll see anything that comes out of the loop of Henle or these collecting ducts is going to get picked up by the vasa recta. So we've got blood vessels ready to pick up anything that's going to make its way out. As we make our way down this descending loop of Henle, a lot of the water molecules are going to get reclaimed, like we said. So what ends up down here at the base, we're going to see a bunch of sodium ions in much higher concentration at the bottom of this loop of Henle. There's still a few water molecules in there, but we did lose a lot of them. And there's still going to be some waste in there, so let me add that. A couple of drops. The cell type changes as we move around this sharp turn of the loop of Henle. When we start to make our way up this ascending limb, we're going to see that the cells permeable to water are no longer there. So water is only going to get pulled out along the descending limb. When we start to make our way up this ascending limb, what we're going to see is the high concentration of salt that's in this filtrate is going to start to make its way out of this ascending limb. It turns out there are cells that line this ascending limb that are permeable to sodium ions and nothing else. So no more water is going to be pulled out. Instead, sodium ions are going to be able to go from an area of high concentration out here, out of the nephron, out here in the extracellular fluid, and into an area of lower sodium concentration. So as we end up going up this ascending limb, we're going to see that a lot of the sodium ions are being pulled out. So the filtrate is becoming more diluted. By the time we get up to this distal convoluted tubule, once again, there's some water molecules in there. There are some sodium molecules in there. Once again, there's water molecules that made it in there. There are some sodium ions that made it to the distal convoluted tubule, even though a lot of the sodium was pulled out. I guess I can make a distinction real quick between this thin segment of the ascending limb and then this thick segment of the ascending limb. It's a side note, and I know I've used a lot of my space to write things on the board. I'll just say the thin ascending limb is a place where sodium is coming out of the ascending limb passively. Sodium is going down its concentration gradient, so it doesn't require any energy for sodium to come out of this ascending limb. However, when we get up to this thick segment, it's now a case where there is more sodium outside of the tube than there is inside the ascending limb. So if we're going to pull any more sodium out along this thick ascending limb, sodium is going to be going against its concentration gradient here. So this is going to be sodium reabsorption that's an active process. It requires ATP, where in the thin ascending limb, it was passive. It didn't require ATP. The thing that we really need to remember is that all along this ascending limb, sodium is getting pulled out, so that by the time the filtrate makes its way up here to the distal convoluted tubule, it's much more diluted. Yes, there's some water in there. Yes, there's some sodium ions in there. We still have the urea waste that's in there, but it's not nearly as concentrated as it was. But we're about to enter this distal convoluted tubule and collecting duct. We know it's lined by these three cell types, principal cells, and the two types of intercalated cells. These are sensitive to different types of hormones, and they can regulate how many water molecules or sodium ions that they hold on to. And there's a consequence, we said, with sodium regulation. It's tied to how many potassium ions we hold on to, kind of indirectly. So to see a little more closely about how these cells operate, I would like to erase this diagram and zoom in on this little segment. I'm just going to take this box and blow it up. We're going to have the collecting duct on one side. We're going to have our three cell types hooked up to the collecting duct. And on the opposite side of that collecting duct, there's going to be a blood vessel, a vasa recta if we're in the collecting duct, or a paratubular capillary if we're along this distal convoluted tubule. So we're going to start with a zoom in of this diagram, and the first tube that I've drawn here is just a zoomed in portion of our, we can either consider this the distal convoluted tubule or the collecting duct. We know hooked up to that collecting duct or distal convoluted tubule were the three cells that we mentioned. The first one is the principal cell. The second one that we can mention is the type A intercalated cell. Then the last one down here we can draw is our type B intercalated cell. We're going to see that all of these cells on one end are attached to the collecting duct, and their opposite end is going to be close to one of those blood vessels, the blood vessels that we were talking about before. If it's along the collecting duct, this could be the vasa recta. Or if it's along the distal convoluted tubule, this would be the paratubular capillaries. Okay, so looking at the details, we've got our three cells drawn that line this collecting duct or distal convoluted tubule. Let's see how each of these is going to operate. I'll start by drawing here on the principal cell, and we can even draw these on the type A and type B intercalated cells. On the side that's closest to the blood vessel, we're going to see these channels called aquaporin 3 and 4. I'll just abbreviate this as AQP. It stands for aquaporin, and I'll put a 3 and a 4 next to it. There are several of these aquaporin proteins, and aquaporin 3 and 4 are always present, and they're little open protein channels that let water molecules easily cross through them. And they're only on the side of the cell that faces the vasa recta or paratubular capillary. So this is aquaporin 3 and 4. Since they're embedded right there on the membrane and they're letting water molecules pass through, we're considering this an active protein. But there's another type of aquaporin that is locked away inside vesicles inside the cell. This aquaporin protein we call AQP2, and it's inactive. It has to be activated, and what we know is coming down the collecting duct and could potentially end up in the urine. Some of the things coming down the collecting duct, we said there were water molecules. We were drawing those as the little blue dots. We said there were still some sodium ions that end up down there in the urine. So we have our sodium, we have our water molecules. But we said selectively other ions could end up down there. And we're going to see the first cell here, the principal cell, has receptors. I'll just use the same color and I'll draw a little receptor. This is a receptor for antidiuretic hormone. Maybe just to take a step back, we know that antidiuretic hormone is, well, it's a hormone, so it's a chemical signal that's traveling through the bloodstream. It makes its way to its target organ. In this case, the antidiuretic hormone leaves the capillary. It comes over here and it docks to one of these principal cells that has a receptor for antidiuretic hormone. This leads to a cascade event that comes over here and activates this aquaporin 2. By activate, we just mean that it carries it over here to the tubal side. It inserts this aquaporin 2. Just put AQP2 there. Now that it's activated, what can happen, let me make a little space here. Now that this aquaporin 2 is activated, it can come into contact with water molecules that were about to be lost in the urine. And water molecules now, they have a way to be reclaimed. They're picked back up by these principal cells, and now there's already a channel for them to make their way out the other side. They go through these aquaporins 3 and 4 into this extracellular fluid for a second, and then they're picked back up into the bloodstream. Remember that the job of this antidiuretic hormone is to prevent water from being lost in the urine, and instead reclaim that water, keep it in the blood vessels. So antidiuretic hormone helps us reclaim water. These aquaporin 3s and 4s, they're always open, but we don't get more water into the cell unless these aquaporin 2s are activated. That's one thing that these principal cells are going to do. There's another thing that is exclusive to those principal cells, and I might need to make a little bit of space in here to mention it. In fact, I'll try and squeeze this in there without making any more space on the principal cell. We're going to see there's also another receptor on the principal cell that is sensitive to aldosterone. We know that aldosterone is also one of these hormones. It was produced by the adrenal cortex, and when aldosterone is moving through the bloodstream, we knew that it was going to affect the kidneys and cause the kidneys to retain or reabsorb extra sodium ions. Let's see how that goes. There is another channel, and here's where I'll need to make a little bit more room. Okay, I just wanted to clean this up to show that the principal cells have receptors for antidiuretic hormone. Antidiuretic hormone, we said, was going to lead to the insertion of these additional aquaporin proteins on the tubal side of the cells that face this distal convoluted tubule or collecting duct, so that any water molecules that happen to be traveling through there now have a way to get picked up instead of lost in the urine. These water molecules travel through the cytoplasm of the cell, out the basal side of the cell through those aquaporins 3 and 4 that are always present, and then right back into the bloodstream. So that's antidiuretic hormone helping reclaim water molecules. We said aldosterone is also a hormone that affects these principal cells. And there is an aldosterone receptor that, if triggered, is going to activate this aldosterone channel. Another cascade event happens if aldosterone is present on this receptor that's going to lead to this aldosterone channel kind of being embedded on the tubal side of this cell. I'll just label it with an A once it's been activated. But this aldosterone channel, it's almost like, it reminds me of one of those revolving doors when you walk into some of the big buildings or like a hotel. Where you're going in one way, but then the door is spinning out the opposite way. In this revolving door, what aldosterone does is it puts this revolving door out here on the distal convoluted tubule. So if there are things like a sodium ion that is traveling through the distal convoluted tubule and is about to end up in the urine, this sodium now has a way to move into the cell. So it helps reabsorb sodium. This sodium doesn't stay there very long. It wiggles through the cell and then right back into the bloodstream. So aldosterone helps reclaim sodium so that it's not lost in the urine, it's reclaimed into the bloodstream. But the side note to that is that that revolving door that we mentioned, for every sodium ion that is reclaimed, that is not lost in the urine, the other side of this is that a potassium ion, let me make a little room for that, a potassium ion is secreted into this tube. So we said sometimes potassium ions end up down there in the urine. And one way that extra potassium can get lost is if aldosterone is working for too long of a period of time. Prolonged aldosterone use is going to hold on to sodium at the cost of getting rid of potassium. Remember, neurons need both sodium and potassium, so long activity of aldosterone can lead to this electrolyte imbalance where you got a bunch of sodium, but you don't have enough potassium. So principal cells, they can help reclaim water, they can help reclaim sodium. These are the main cells that are affected by antidiuretic hormone and aldosterone, the main hormones that we mentioned that affect the distal convoluted tubule and collecting duct. But there are two other cells here to talk about, and they are going to play a role not in holding on to water or sodium, but instead they're going to play a role in helping regulate the pH of the bloodstream. These two cells are really concerned about hydrogen ion concentration, the pH of the blood. we're going to see, and I think I gave this away earlier, if the blood is too acidic, then type A intercalated cells are going to kick into gear. Let's see what that looks like. If the blood is too acidic, that just means that it has too many of these hydrogen ions in it. I'll draw 4 of those hydrogen ions, and then I'll draw just 1 of those bicarbonate ions. So if we look at the blood right here, the blood is way too acidic. There are too many of those hydrogen ions. And the type A intercalated cells can do something about this. They can regulate the pH. And if we dive inside of it, here's what's going on. And once again, I'm going to give myself just a little bit more space. Okay, so now I've got a little bit bigger type A intercalated cell, so we can write what's going on in here. There is cytoplasm inside these cells, and if there's water inside the cytoplasm and carbon dioxide, we said those two things mix. Water and carbon dioxide are going to combine and form this molecule that we call carbonic acid. There's our carbonic acid. It's an unstable molecule, so it's going to break apart into 2 components. One component is a hydrogen ion, and then the rest of it, the other component, is this bicarbonate that now gets a negative charge. The bare proton with a positive charge, this hydrogen ion, goes one direction, and the bicarbonate, this negatively charged ion, is going to end up going the other way. This cell splits this carbonic acid, and we're going to see that it reabsorbs this bicarbonate. So it's going to help cancel out these free hydrogen ions. These two things cancel each other out. So by pumping in extra bicarbonate, this is going to help neutralize the pH of the blood. And the other thing, if this carbonic acid breaks down where half of it, the bicarbonate, goes back into the bloodstream to neutralize the pH of the blood, the other thing, this hydrogen ion gets kicked into the tubes. Gets kicked into that distal convoluted tubule and is going to end up down here in the urine. We said sometimes there are hydrogen ions in the urine. The urine will be acidic if the blood is acidic. And that's how the kidney is compensating for the blood being too acidic. It's putting in extra bicarbonate ions to neutralize that acid, and out of the other side, it's getting rid of these hydrogen ions in the urine. There's a side note that I need to add even to this. We're going to see that there's something that happens right here that when one of these type A or even type B, one of these intercalated cells gets rid of a hydrogen ion, it's getting rid of a positive charge, so it has to fill that void by picking up a positive charge. What it picks up is one of these potassium ions that might be out there in the filtrate about to get excreted in the urine. That potassium ion gets reabsorbed, and it's going to end up making its way through the cell and into the bloodstream. That's the side note. Just to say, type A intercalated cells, they neutralize the acidity of the blood by adding this bicarbonate, but what they also are adding is this potassium ion. We mentioned, or we may not have mentioned before, but there is a tight regulation on how many potassium ions, like the concentration of potassium ions in the bloodstream. Too few of these potassium ions, it's what we call hypokalemia. And if there are too many of these potassium ions, it's hyperkalemia. If type A intercalated cells are operating for a prolonged period of time, it can lead to hyperkalemia, an excess of these potassium ions in the bloodstream. And you're doing that because you're actually getting that because these cells are trying to compensate for the blood being too acidic. They're getting rid of those hydrogen ions and just replacing it with a potassium ion. Let's jump down to the type B intercalated cells, and we're going to see that the type B intercalated cells, they do pretty much the exact same thing that the type A intercalated cells do, but it's just reversed. It's flipped around backwards. These type B intercalated cells, they operate when the blood is too basic or too alkaline. If we want to visualize that, that would mean that there are fewer of these hydrogen ions than there would be the bicarbonate. So in this case, we have too much of the bases that are here. There's not enough of those free hydrogen ions. That the blood being too alkaline like this is also problematic. No problem. Type B intercalated cells are on the scene. The same reaction is taking place inside these type B intercalated cells. So we've got water and carbon dioxide that are going to form this unstable molecule, carbonic acid. And like we said, the opposite thing is going to happen this time. As this disassociates, the proton, this hydrogen ion, is going to end up being dropped into the bloodstream. So those cancel themselves out, and as more of these hydrogen ions are reabsorbed into the bloodstream, that's canceling out all of those bases. So now we've returned the blood back to our neutral, our normal range of pH. The other side of this, we said, was the bicarbonate. We don't want any more of that in the bloodstream. This is going to go the opposite way and get secreted into the urine. And like I said, that makes sense. If the blood is too alkaline, if the blood is too basic, then we're going to see that the urine is also going to be alkaline or basic, because you're trying to get rid of these excess because you're trying to get rid of these excess bicarbonate ions. The other part of the story is true. The side note that we added, if this cell is going to get rid of one of these positively charged ions, it's going to need to replace it with a positively charged ion. It picks up one of these potassium ions from the bloodstream. So type B intercalated cells, they reabsorb hydrogen into the bloodstream, but they're going to secrete potassium. This potassium is secreted from the bloodstream through these intercalated cells and right into the urine. So type A intercalated cells are going to reabsorb potassium, reabsorb that potassium into the bloodstream. Type B intercalated cells are secreting potassium out into the urine while they're reabsorbing hydrogen. So there's always a swap of these positive ions. That is sufficient. We can see that type A and type B intercalated cells really aren't affected by hormones like aldosterone. They're really just regulating the acidity of the blood, getting rid or holding on to those hydrogen ions. Then the side effect of holding on or getting rid of those hydrogen ions is that you're also either holding on or getting rid of potassium ions. Okay, enough of that. Let's catch up with some of the slides. Okay, now that we've got that drawn, we can catch up with some of these slides. At the beginning of our diagram, we were looking at this Bowman's capsule where filtration occurs, and we said if you were a ion or a glucose molecule or a water molecule, if you're small enough to fit through the little pores, then you're going to be filtered out into this Bowman's capsule. And if we look a little bit closer at Bowman's capsule, we can see here is this Bowman's capsule. It's sometimes referred to as the renal corpuscle. But to be clear, this renal corpuscle is two things. It's the glomerulus, which is the capillary itself, and then it's also Bowman's capsule. Bowman's capsule and glomerular capsule are the same thing. That's describing these epithelial cells. There's two layers to the Bowman's capsule or glomerular capsule. There's this parietal layer of cells. That's a simple squamous layer of epithelial cells. And then you can see the visceral layer. Those are the nuclei of the visceral layer. These cells are called podocytes, and they're directly touching the surface of the glomerulus. We've got our larger afferent arteriole taking blood into the glomerulus, and then this smaller efferent arteriole draining it away. The other thing that you can see is that you notice as soon as we get away from Bowman's capsule and into this proximal convoluted tubule, the cell type changes. It goes from simple squamous to simple cuboidal. And you can even see that the apical surface of those cells have a bunch of microvilli, and that's just trying to increase surface area so reabsorption can occur along that proximal convoluted tubule. Here, if we zoom in even closer to this glomerulus, you can see those pores, those fenestrations we said that were present in the glomerulus. That's where we found fenestrated capillaries. There are the fenestrations. In addition to those pores adding to filtration rates, we're going to see that the podocytes themselves can also adjust filtration rates. They can change their shape. Here's the nucleus of the podocyte. It has these long appendages, and if we look at the tips of those appendages, we can see these little finger-like projections. Those are the podocytes. Those are the feet processes of the podocytes. And if we could zoom into those little feet processes there, if I could take a cross-section through all of those little feet processes, it would look like this. Those are the little slits in between each of those little foot processes. And this top picture is showing us a picture where each of these little foot processes are relaxed. And so they're standing straight up like this. And when they're standing straight up, there's little gaps, these spaces where substances could filter through those spaces. So when the podocytes are relaxed, it widens these filtration slits and filtration increases. This bottom picture shows that those podocytes, they have actin and myosin filaments in there, and they can contract. If they contract, instead of them standing straight up, it's like they squat down and they kind of widen out. So you can see at this bottom picture, those podocytes have widened out, and when they do that, they close off those filtration slits. So in their contracted state, they decrease filtration rates because they close off those little filtration slits. So once again, there is another way in which Bowman's capsule can help regulate filtration rates by either relaxing or constricting the feet processes of the podocytes. Relaxed increases filtration rates, contracted decreases filtration rates. So filtration happens in Bowman's capsule, and then we said in the proximal convoluted tubule is where anything that can be reclaimed, like water and sodium and glucose, is going to be reclaimed. This is what we refer to as tubular reabsorption. We're trying to reclaim things that were already in the bloodstream. They came out temporarily during filtration, and we're going to try and pick them right back up. What was on the board earlier showed paratubular capillaries and vasa recta. And I know there's a lot of words on this slide, but the only thing I really want to point to on this particular slide is that we can see a nephron. This is a shorter nephron, what we called a cortical nephron, whereas this is a juxtamedullary nephron. It's got a really long loop of Henle that goes down into this medulla region of the kidney. This is just showing us, the main thing I want to see with this slide is that the blood vessels that surround that loop of Henle are called the vasa recta. And they're going to be picking up anything that comes out of that loop of Henle. Just like the paratubular capillaries up here, all of those purple blood vessels, notice how the paratubular capillaries are just surrounding the proximal and distal convoluted tubule. They're not around that loop of Henle at all. We said those paratubular capillaries are reclaiming anything that gets pulled out of the proximal convoluted tubule or distal convoluted tubule. So those are the blood vessels, like we said, reclaiming things that are coming out of the nephron. Along that proximal convoluted tubule, this is where all of the glucose molecules get reabsorbed. So there should be 100% of glucose molecules that get reabsorbed along that proximal convoluted tubule, so that none of them end up in the urine. That would be wasteful and not good for our cells that could use that as a fuel source. So 100% of the glucose is reclaimed, and most of the water and most of the sodium is reclaimed, almost 99% of it. But we can see some of the water makes its way through that proximal convoluted tubule, and some of the sodium makes its way through the proximal convoluted tubule. And again, some of that's going to end up in the urine. The other stuff that we see that is reabsorbed at the proximal convoluted tubule is urea. But once we move through this proximal convoluted tubule, we find ourselves down this loop of Henle. There's the descending limb of the loop of Henle, and then the ascending limb of the loop of Henle. And I guess I'll just point to it here. We had it up on the board earlier. We need to remember that all along the descending limb of the loop of Henle, that blue arrow is showing us that the descending limb is permeable to water. So water is going to be making its way out of the loop of Henle, leaving behind more concentrated filtrate. So down here at the bottom of this loop of Henle, we have the most concentrated filtrate. It's Doesn't have a lot of water in it compared to all of the sodium ions and waste. But as we start to make our way up this ascending limb, we said no more water is going to be pulled out, but instead sodium ions are going to be pulled out. And as more and more sodium ions are extracted from this ascending limb, it leaves behind more diluted filtrate, more diluted what we are about to call urine. So when we get into this distal convoluted tubule, we see things like water and sodium in lower concentrations. There's also waste products and other ions that are present in there. And depending on what hormones are going to be moving through the bloodstream, we'll see the distal convoluted tubule and collecting duct behave differently. We know the cell types. They can either be reabsorbing things like sodium or water, or perhaps they're secreting things like potassium. So we'll talk about this connection between hydrogen ions and potassium ions. The thing to remember is that aldosterone is going to lead to the reabsorption of sodium. We see aldosterone putting these additional channels that allow sodium to not be lost in the urine, but be picked up by the blood vessels. We see antidiuretic hormone is helping the reabsorption of water molecules, so that antidiuretic hormone put additional aquaporin proteins on the tubule side of these principal cells, gave additional channels for water to not be lost in the urine, but to be reclaimed by the principal cells and then eventually back into the blood vessels. Aldosterone reabsorbs sodium. Antidiuretic hormone, also called vasopressin, reabsorbs water molecules. But there's secretion involved in both of the two. We said aldosterone, even though aldosterone is reabsorbing sodium, it's secreting potassium. Even down here, those intercalated cells, we said they're either getting rid of hydrogen ions, but if they're getting rid of hydrogen ions, they're reabsorbing a potassium ion, or vice versa down there in the type B intercalated cells. Here's the sketch. In fact, this is kind of the theme of the sketch that I had on the board originally, where you can see parts of the nephron. This descending limb of the loop of Henle, we said, is permeable to water. So notice how the filtrate goes down this descending limb. Water gets pulled out and it leaves behind this really concentrated urine. We can see the concentration of solutes is much higher than it was before we went down this descending limb. So now that we've pulled all the water out, we've got just a bunch of sodium ions and water and a few other ions down there. Along this ascending limb, we're going to see that it's permeable to the sodium. At first, sodium comes out passively, and then up here at the very end, sodium is going to have to be pulled out actively. But the main thing is that all along the ascending limb, sodium is getting pulled out. And you can see what that leaves behind is more diluted urine, more diluted filtrate. So there's still some sodium ions, there's still some water molecules in this stuff that is about to be urine. And we said if antidiuretic hormone is present, then there's additional channels for water to be reclaimed and pulled back into this vasa recta rather than being lost in the urine. There are those aldosterone channels doing the revolving door thing that I mentioned. It prevents sodium from being lost in the urine. Here's that sodium being reclaimed and pulled right back into the blood vessel. But every sodium that it reclaims, it gets rid of a potassium. So that's the double-edged sword with aldosterone. This is just a sketch. Let me try this again. Here is the detailed slide of this principal cell sketch that I had put on the board. What we can see, and also you'll notice that the diagram here on the overhead is a mirror image of what we have drawn on the board. And maybe it's good to see it both ways. We've drawn our capillary, our blood vessel over here to the left, and our nephron tube to the right, and that's the opposite of the slide. Look at how the nephron tubes are over here. So if we want to reclaim extra water, those type 2 aquaporin channels are inserted, like we said, on the tubal side of these principal cells, so that any of that water that was about to go down the collecting duct and end up in the urine isn't going to end up in the urine. It's going to be picked up by these principal cells, and those type 3 and 4 aquaporin channels, they're always present on the basal surface of this cell. So if water molecules can get in, they have a way to get out and right back into the bloodstream. What activates, we said, those aquaporin 2s is antidiuretic hormone. It docks to the antidiuretic hormone receptor, activates these aquaporin 2s, and now we have even more channels for water to make its way through and be reabsorbed rather than lost in the urine. So there's vasopressin. I'll let this picture serve as our showing how aldosterone works, just that revolving door. For every sodium that's reclaimed, a potassium ion is secreted. So there's aldosterone, there's antidiuretic hormone, and then that just leaves these intercalated cells. Here's the detailed picture of our type A intercalated cell. And really the heading up here on the screen is everything that we need to remember. What we need to remember about type A intercalated cells is that they are activated when the blood is too acidic. So what they do is they secrete, they get rid of these hydrogen ions because we already have too many of those. And they reabsorb the bicarbonate, that negatively charged ion, because we need more of those bicarbonate ions. We want to get rid of those hydrogen ions. That's the main thing. But we said the side note to that was that in order to get rid of one of these hydrogen ions, to give up a positive charge, you got to replace it with a positive charge. So for every hydrogen ion that we secrete, there is a potassium ion that is reabsorbed. So type A intercalated cells secrete hydrogen. They get rid of that hydrogen, but the downside is that they reabsorb potassium. And they also reabsorb that bicarbonate, trying to neutralize the acidic blood. That's why we said if the blood is acidic, the urine is also acidic. Second slide shows us those type B intercalated cells, and they're doing the opposite of the type A intercalated cells. These would be active if the blood is too alkaline. We don't have enough of the hydrogen ions in the bloodstream. So you can see the type B intercalated cell actually reabsorbing one of these hydrogen ions into the bloodstream. It's swapping that hydrogen ion for another positively charged ion, that potassium ion again. So type B intercalated cells are reabsorbing hydrogen but secreting potassium. That potassium would end up in the urine, just like that bicarbonate. This bicarbonate, we already had too much of that in the bloodstream, so we don't want to add more. We want to get rid of that excess bicarbonate. So if the blood is too basic or too alkaline, the urine is going to be also basic or alkaline. And then the final thing that we see here is, I think this is one of the sketches in the lecture handout for urinary system. It shows a close-up picture of Bowman's capsule. And you can see Bowman's capsule kind of wrapping around the glomerulus. We even see the kind of the darker staining cells there. Those are the podocytes that surround the glomerulus. They can adjust filtration rates. About filtration, we said podocytes can adjust filtration rates, but there's also this built-in mechanism that we had mentioned earlier that's built into the kidneys that can sense if filtration rates drop too low. And they can respond by doing several things that boost filtration rates. If this isn't something that we put on the board before, I know we've put on the board before that blood volume is directly related to blood pressure. But now that we're talking about kidney and filtration rates, we can add one more thing. We can say blood volume and blood pressure. These are all directly related to filtration rates. So all directly related to one another. If I do something to boost my blood volume, then that's going to boost my filtration rates. My dad does this all the time. He'll sit there and drink like a case of Dr. Peppers. He's increased his blood volume, and now he's going to the bathroom a lot. His kidneys are producing a bunch of urine because blood volume is increased. That increases blood pressure, and that increases that pressure in the glomerulus. So more filtrate is coming out. More urine is being produced. Again, podocytes can adjust that, but the more problematic scenario would be if filtration rates drop too low. Now cells are drowning in their own waste, and they can't get this waste off. They can't produce urine. So there is a collection of cells that line this distal convoluted tubule that have little receptive ends that can sense the filtration rates in this distal convoluted tubule. And if the filtration rates drop too low, they can respond by stimulating a group of cells called mesangial cells to produce renin. Here's where it should sound familiar from something that we mentioned earlier in the semester. We mentioned this renin, angiotensin, aldosterone system. We said that if blood pressure drops too low, right? If blood pressure drops too low, that's the same thing as filtration rates dropping too low, and the kidney can respond by producing renin. And renin does things to boost blood volume and boost blood pressure. Well, renin is produced right there by the mesangial cells. This is the first time we actually got to look at the cells that actually produce renin. They are told to produce renin by these macula densa cells. Those are the columnar-shaped cells that line a portion of the distal convoluted tubule, specifically the part of the distal convoluted tubule that makes contact with the mesangial cells. Unlike I drew on the board, I guess I should also point this out. I drew a nephron on the board that was basically stretched out, and that is not an accurate representation. It would be more accurate if I could have taken the distal convoluted tubule on my diagram, peeled it off of the board, and then overlaid it on top of the proximal convoluted tubule. Here, notice that this is Bowman's capsule. As we go away from Bowman's capsule, this is our proximal convoluted tubule. It eventually makes its way down into this descending limb. Then we make our way back up this ascending limb. And then the ascending limb gives way to the distal convoluted tubule. But look how this ascending limb, where the ascending limb ends and the distal convoluted tubule begins, is right when that tube passes next to the glomerulus again. If you look here, you can see that this ascending limb, what is about to be called the distal convoluted tubule, actually passes between the efferent arteriole and afferent arteriole. Here's our efferent arteriole, and this is our afferent arteriole, and there's that distal convoluted tubule kind of splitting the two blood vessels themselves. It gets right next to that glomerulus, and it's for a reason. It's so these macula densa cells can communicate if filtration rates drop too low. They can communicate to those mesangial cells to release renin. One of the things that renin is going to do is it's going to cause these granular cells that are right there surrounding that afferent arteriole. These granular cells, they're smooth muscle cells. They're going to relax. And that's going to be one of the things that allows more blood flow into the glomerulus. The other thing we'll see is the smooth muscle around the efferent arteriole is going to contract. So more blood flow into the glomerulus and less blood flow away. That's going to lead to a buildup of pressure immediately in the glomerulus, boosting filtration rates. Key thing from this slide is that we've now found the cells that produce renin. Those are the mesangial cells. Macula densa cells tell them to produce renin if needed. That renin, amongst other things, is going to cause these granular cells to relax. Efferent arteriole is going to contract. Everything that it does is to boost those filtration rates. So we had seen this before, this renin, angiotensin, aldosterone system. We don't have to remember the details. This is the last slide, and it's just kind of a review slide, because now we've seen the cells in the kidney that produce renin. All renin did was activate a protein that was in your bloodstream already. It was just in its inactive form. We call that angiotensinogen. It doesn't do anything in this inactive form, but when it comes into contact with renin, it is converted from angiotensinogen into its active form of angiotensin 1. Even angiotensin 1, even though it's active, it doesn't do a whole lot. It has to pass through the lungs and get converted a second time into angiotensin 2. What converts it from angiotensin 1 to angiotensin 2, we said, was this ACE enzyme. It stands for angiotensin converting enzyme. It's found in the lungs. So as blood passes through that pulmonary circuit, through the lungs, and then eventually back into the left side of the heart, we're going to see it's now been activated into angiotensin 2. That's really where all the magic happened. We listed several things that angiotensin 2 was responsible for, and all of these things boosted blood volume or boosted blood pressure. We can see that being thirsty and drinking a bottle of water is going to increase our blood volume, which helps increase blood pressure. There is arterial constriction. So if the volume in the blood vessels decreases, that's one thing that can cause pressure to increase as volume decreases. The other thing that we can do to increase blood volume is to retain more water. So vasopressin or antidiuretic hormone is something that is stimulated to be released by this angiotensin 2. And then the final thing angiotensin 2 was doing is going to the adrenal cortex, getting that adrenal cortex to release aldosterone. And we know aldosterone over here is going to allow these kidney cells to reclaim extra sodium. And that kind of goes hand in hand. We said that water molecules, they like to follow around wherever sodium goes. So antidiuretic hormone and aldosterone have an enhancing effect on one another. If sodium is being reabsorbed, it's going to be easier to reclaim or reabsorb more water. Both of those are boosting blood volume, which boosts that blood pressure and gets those filtration rates back to operable range. So again, we don't really need to remember the details of angiotensin-aldosterone system, but what we do need to know now is the cells that are responsible for production of renin, kind of right at the very beginning, and then eventually the cells at the very end that are responsible for reclaiming extra sodium, the principal cells, or reclaiming extra water, once again, those principal cells, or the cells responsible for regulation of the pH of the blood, the type A or type B intercalated cells. So that's it. Hopefully that's been helpful. We will see a further PowerPoint on electrolyte balance on Thursday, and we'll do an in-class case study that will present us with scenario-based questions about this so that we can get some practice with it.